Dear colleagues, my name is Ruslan. I'm group leader at the Institute for Regenerative Medicine at the University of Zurich in Switzerland, and it's a pleasure to present you our newest project in the lab about how to generate quantitative, high-quality behavioral data from stroke mice with the help of deep learning. Now, as most of you know, behavioral evaluation in preclinical animal models is extremely important, and still many labs only use very superficial behavioral techniques that may not reveal the full complex information about functional deficits or recovery. So when looking at a mouse performing a free walking runway, it's hard to identify specific alterations in the gait. We wanted to change that. Here we took advantage of the recently established software by Mattis and colleagues called DeepLabCut. The software enabled us to track all the relevant body parts and joints of the mouse in each frame of a video and to build a comprehensive locomotor profile after stroke. So how does it work? We performed the behavioral tasks and recorded the animals from three perspectives. These videos were then imported into DeepLabCut. There we extracted frames and labeled our regions of interest, like the joints, the body parts, and so on, and trained the software to recognize it. These trained networks were then applied to new videos and provided us with coordinates X and Y that we used as an input for our analysis to identify biologically relevant gait differences between stroked and non-stroked mice. To verify the accuracy of the networks, we checked first the cross-entropy loss and the likelihood for correct labeling. And with our setup, we achieved a cross-entropy loss of less than 1 in 1,000 frames and had an average likelihood to label each body part correctly of more than 99%. So we could use almost every data point that we have generated. Next, we introduced the phototrobotic stroke in our mice, targeting specifically the sensory motor cortex, and followed the animals during a behavioral performance at regular intervals for 3 weeks. A successful stroke was confirmed by a severe reduction in blood perfusion levels using laser Doppler imaging. And later, also in a histological analysis, where we measured a stroke size of around 1 to 1.5 cubic millimeters. We began the motion tracking analysis by assessing the overall gait from the bottom perspective at baseline and after injury. Individual steps were identified by the movement speed of each limb between the frames, and the footfall pattern, for example, showed a typical gait synchronization of opposing front and back paws in uninjured animals. Now, if we look at the step cycle in injured animals, we see that the pattern was severely altered. Then we compared the kinematics of the fore and hind limb joints from the side perspectives. As an example, we identified alterations in the average height of the fore and hind limb joints that was, as expected, more prominent on the contralateral left side, with a partial but incomplete recovery over time. Importantly, there was also some late changes in the ipsilateral side that may be an indicator of compensatory movements. To next understand the individual importance of the, in total, more than 100 measured parameters during our analysis, we applied the random forest classification throughout the entire time course. We were able to predict the acute injury status with 90% accuracy and long-term deficits at the accuracy of 85% using a confusion matrix. Interestingly, the overlap between the most important parameters in acute and chronic time points was only 20%, further confirming the complexity of recovery over time. Next, we considered whether neural networks can also be applied to conventional behavioral tests to detect, for example, fine motor deficits in a ladder rung test. Tracking the fore and hind limbs during this ladder rung recordings enabled us to identify stepping errors. As expected, the increase of the overall rate in injured animals was higher compared to intact controls. We then cross-verified these results or these error rates in injured animals compared to blinded observers. And although we did not detect any differences in the scoring accuracy to the gold standard, the manual assessment, we saw that a manual assessment required 200 times as much time compared to the DLC-assisted approach. As the last step, we benchmarked DLC tracking performance against popular functional tests to detect stroke-related functional deficits. And in summary, we can say that only the most sensitive conventional tests were able at all to detect long-term impairments after strokes. But these tests were often very tedious and still provided very few readouts, whereas DLC-assisted tracking that we present here provided many biological parameters. The setup reduced analysis time while minimizing the observer biases during the evaluation to generate quantitative and reliable behavioral data. Finally, I would like to acknowledge the many amazing people working on this project, the funding bodies, and of course you for your attention. Thank you.